Hi guys. I didn't have a privilege to attend worm meeting for a while, and thus far it was really great, and I'm glad to be here. With that, some regrets we have later in life and some instantaneous, so I regret that I didn't go to buy the lovely Iron Maiden t-shirt while presenting the, it could have been much more exciting way of starting this talk, but you know. So just to say, I'm not working in Celegans lab. I'm working in EM facility at the University of Lausanne, but I have a background and affinity to Celegans since my master's, even before, and I do develop different techniques or approaches that can make it easier, and we are all here, most of us are here, because we love this amazing creature, model system, whatnot, but it's not universal. So if you are an EM person, most likely you hate them. And, and you hate them for the very same reason C. elegans people like them. And it's basically because, well, they're small, transparent, they have many different stages, which on its own kind of a different animal. You need to process them one by one. The region of interest, like an organ can be 1 or 2 cells. So yeah, and you've heard, so also the way to prepare them, it's not evident if you compare it to different EM techniques or model organisms that used for or analyzed by EM, and high pressure freezing, sectioning, and eventually data interpretation can be quite tricky. So the cornerstone and basically the curse of TM is mainly, or at least people refer to sectioning, ultrasyn sectioning, because it's tricky. So we use the diamond knife and the ultramicrotome, and you saw we transfer it. It's not 100% to scale, but that's how the grid, that's the compared to the knife, which is small on its own, the grid is really tiny. So that is the step that takes a lot of persistence and a lot of frustration sometimes. And that's why different types of techniques started to emerge actually to tackle after all these steps. So you've heard, that's not that one. Yeah, so it's possible, and you saw from Mia and whatever work can do it, but not everyone. But you can collect really a lot of sections on one slot grid, but it takes a lot of practice, and especially if you need a consecutive, like hundreds, thousands of sections, it becomes very complex task. So one of the things, this array tomography technique proposed by Christina Mikiva from Stephen Smith lab, was to actually avoid this step of picking the sections on the grid. And that's why they started collecting the sections on a glass slide using this larger boat knife. Since they published the technique, different other techniques emerge. I use a slightly different knife, but the idea is also, and instead of glass slides, I use bits of wafer. I will show you later what it means, but just to give you an idea how much work you will need to invest. So if you have this relatively modest size of a section, and you, let's say, manage to put, given the size of the grid, 3 sections per grid, you will need plenty of sections, of grids to collect 100 sections. If you are using a wafer, right, you can quite painlessly collect the same amount of sections on one wafer, and not without cursing everything, you know, is dear or not to your, in your life or in your surrounding. So that's basically a composite image of a grid with some section, with 3 sections, and those are actually these 100 sections transferred on this bit of silicon wafer. So that was part, so the knife, the idea of this kind of large basin diamond knife for which where you can put the wafer, so the wafer is this square rectangular shape that is here. Here you see the sections. Those are consecutive sections that stick to one another. And that's what we call ribbons or arrays. And basically that's your most important tool, right? This eyelash on a pick. And that can be a bit tricky to move, not tricky, you need to have patience to learn how to move it inside the basin, but when you do, life is good, because after you can align them as you wish, and I probably cannot make it faster. I cannot, I wanted to make faster, but trust me. Yeah, it either does that. So basically when you align the ribbons over the wafer, you can draft water. The water would drain entirely, and then your ribbons will stick on the wafer. So the movie is supposed to go all the way to show how it goes down, the water goes down and how the section dry, but that's schematically, that's what it does. And yeah, indeed, I think that this detachment and arrangement are basically the most difficult part, but if you compare it to serial sectioning and trying to align and follow the sections on the grids, it's really great. It's not the difficulty. So basically, when you transfer, you can, because those sections that you can you generate with ultramicrotom, you can decide how thick the section will be. So basically, here I just named the different types of different thickness of sections. And as far as and it all depends what you want to do with it, because sometimes people say it's important that they remain straight, and it's important that they will be less than 50. It all depends what question you want to address. And you saw that, so whatever we do with this on large surface methods, we do it inside the SEM, scanning microscope chamber. And the idea is that it's a bit different. It's not the same as TM. It's not the penetration of the beam through the section. It's actually what the detectors can collect. And if you start doing that, or if you have a fib or scanning block phase, for sure you can do array tomography. If not, it's basically just check the parameters of your machine, and it's important, not to say, oh, it doesn't work, that's all. There are so many things that you need to play with, and it will work. So basically, you can either have an automatic or manual acquisition of the sections on the array. I'm using the FEI maps, in which you can actually, so that's the wafer, and that's the map of the same wafer inside the microscope. And then when you find places, the regions of interest, you can decide what you want to collect, in which sequence, and how it will look. So that's eventually you will get your 3D if that's what you're looking for, or actually any size of the sample. So basically what I like to use is this lateral screening approach, which you don't check everything, you just sample in each ribbon. You go and sample one section, and then because you know the anatomy, you know what you are looking for, you actually can see which parts are, let's call them obsolete, and which parts are important, because here it's a gonad. Let's say I was interested in that. And that makes the entire experience much more pleasant. You don't suffer with in and out the grid from the TM. Whoever knows what I'm talking about, whoever not, just trust me. And another huge thing about array tomography is actually that you can change very easily the orientation of sectioning. And most of us are used to this transverse orientation, right, the sausage way. And that is not very intuitive, especially for people who use fluorescent microscopy, and it's basically not how we can directly interpret the data. However, if you have a longitudinal section, you already can obtain much larger area, and also it will be in a familiar orientation. And then there is this part of tiling, right, where you can actually acquire many different images with high resolution, stitch them, and then you can zoom in and out and have all this information both on the very tiny level and on the global level. And that's one of the benefits of array tomography, because actually in scanning block phase and in the FIB, whatever you imaged, it's gone. You cannot go back and say, let's see, let's check it again, let's how it goes. And so yeah, as basically I started with array tomography because it was so appealing for the correlative microscopy to find a signal and then in fluorescence and then superimpose it and see what where it localizes in different on the ultrastructural level. And actually, so yeah, one of the problems, so the labeling was fine, the procedure was fine, it takes a while, but when you want to preserve GFP, sometimes, or fluorophore, sometimes it works, sometimes it doesn't. So I did several, so this was the project with Mihoko Kato from Steinberg Lab. They wanted to see the leader cell of male gonad. So the conditions were right, and I managed to find the cell and 3D and total correlation, and that was fun. It was very satisfying to, even though it was kind of a long project, but then that was basically a turning point for me, because I think that the idea of having 3D correlation between light and electron microscopy doesn't make much sense, because we know what we are looking, as long as we know what organ we are analyzing, the fluorescence is irrelevant because the ultrastructure would be already good enough. So that was in project with Wendy and Max. They had quite an ambitious project involving different sensory neurons, and actually the dimorphism between the male and hermaphrodite. And at first we thought to do something like correlative, but then because they had so many different conditions, and for each condition it was important to see that more than one animal is behaving the same, right? So I do the flat embedding. I think it's imperative for this kind of sectioning, or the approach that we want to analyze a particular stage, particular sex, and analyzing the animals that we know where they come from or what sex they are. That was quite important. And then eventually it was a great approach. I mean, it was so, even though I worked on this type of procedure and I did a lot of array tomography before, these experiments were a revelation for me as well, because I managed to do plenty. So those are just 4 examples, but for each of these genotypes, I have acquired the stacks, around 100 sections, and actually it's quite statistically significant. It's not one animal, and it's not like only 2 mutates. So I was really glad that we managed to do that. And actually, embryos, I learned that with this approach, embryos is actually even easier than the worms, because then what I do, I just collect a lot of worms, of embryos from the stage that I want to get, and then I just blindly cut into the into different blocks, and I can find the stage I'm interested in after I section. And it's so easy because of the known anatomy, and it's not. And you can, because I will collect eventually the entire stack, the orientation is not really important, or not really important to interpret the data. And that was the project that I did with Anthony Santella and Jerome Bao. And the idea was, so we had several data sets with both Fib and array tomography embryos, and instead of doing like direct correlation when you have fluorescence and then you try to figure out what is what, we wanted to use the algorithm that Bao Lab developed for the live imaging, and then actually retranscribe it to the EM stacks. So that was quite an impressive project. So yeah, it's published if you are interested. And I just wanted to, so all techniques that we showed you, it's really, yeah, no, let me just, so I do think that array tomography is a great technique for C. elegans. It makes things much easier, especially if you need to analyze. If you are not doing something that is a global reference, for the day-to-day work, it's a really great way of analyzing the samples. And basically, between all the strategies that we showed you, you can do a lot with either of them, or double, for the same question. It all depends, the choice depends on the size of your region of interest, the speed you need the result, the resolution you require, both X, Y, and Z, and if you have money and manpower. So basically a lot of this is eventually, stops where the money comes to the game, but I mean, you need to obtain the results, so just take the technique that can do it the best. So basically, I took a bit more, sorry, and thank you for not moving me. But now we can open the questions, whatever you have, like, for regarding our presentation, but we didn't mention any of those, and those are basically parts that indirectly related to electron microscopy and the problematics of electron microscopy, but not only for C. elegans. So yeah, just to give you an idea, I just listed them. Thank you very much.